So welcome everyone who's logged in, and as you're logging in, I'm going to give a very quick background into what this lecture is all about, why it's so special. First, I want to give a really warm welcome to Dr. Doherty for being here, for accepting. He's coming to us right now live from the other side of the world, so it's great to be able to have him, not in person, but at least have him come to speak to us as in person as possible. Special thanks to the Student Association for taking the initiative to organize this really great event. It's always a pleasure to be a part of and to see it. For those of us who don't know the IRCM, you might be logging in just because you saw the announcement and don't really know who the IRCM is. We are a independent research institute in Montreal that was founded over 50 years ago with the purpose of facilitating translational research that's directed really by clinical questions. I want to take a minute to talk about the people, though, for whom this special guest lecture is about and dedicated to. So Marie and Willie Chrétien were married on May 27th in 1909, and they were the parents of a very large family. They always put education as their first priority for their children, even back then, and for all their children, even their daughters and their sons. They ensured that all 9 of them, I repeat, 9 of their children, obtained college educations, and 6 of them obtained university degrees. While Willie Chrétien spent all 50 years of his working life as a mechanic at the local paper mill in Shawinigan, Marie Chrétien took charge of most of the family responsibilities. Marie and Willie Chrétien would have been very proud to learn that the IRCM students are hosting Professor Doherty for the 2021 lecture that is named in their honor. Now I'll leave it to Sophie to introduce Dr. Doherty.
Thank you. So I'm Sophie. I'm the president of the Student Association, and we're very honored to receive Dr. Doherty for this great seminar series. So I'll just give a bit of background into his achievements. So Dr. Doherty received a bachelor's degree in veterinary science in 1962 in Australia at the University of Queensland, after which he became a veterinary officer in the Department of Agriculture and Stock and spent some time studying the epidemiology of infectious diseases in cattle and pigs. So the specific study of bovine leptospirosis was written up as a master's thesis and published in local journals. He then sailed to Scotland on a, quote, very slow and cheap ship to pursue a career in the Department of Experimental Pathology at the Morden Research Institute, where he later enrolled as a graduate student for the University of Edinburgh. There, he focused on the tick-borne flavivirus and even accidentally injected himself in the finger with the virus, eventually publishing the results of the ensuing serological tests as a letter to the Lancet. Later, he left his permanent research position to work as a postdoctoral fellow back in Canberra. His studies, along with Rolf Zinkernagel, if I'm pronouncing this right, revolutionized the immunology world and theorized the single T-cell receptor altered self principle. They discovered that for killer T cells to recognize infected cells, they needed not only to recognize an antigen, but also the major histocompatibility complex at the surface of these cells. This eventually earned them a Nobel Prize in medicine. At the time, though, he took an associate professor position at the University of Pennsylvania, where he worked on the influenza model, rabies studies, and research on multiple sclerosis. He now works part-time conducting research at St. Jude's Children's Research Hospital in Memphis, Tennessee, and the rest of the year in the Department of Microbiology and Immunology at the University of Melbourne. His study focuses mainly on the T-cell response to viral infection. His work has yielded numerous groundbreaking publications in journals such as Nature, Science, and Immunity. He's a member of the Australian, British, American, and Russian Academies of Science, has more than 20 honorary doctorates, and even has his face on postage stamps in Australia. In addition to his Nobel Prize, he's won several prestigious awards, such as the Lasker and Gardner Foundation Awards. Alongside his research work, Dr. Doherty is also the author of a number of books, such as The Beginner's Guide to Winning the Nobel Prize, or Light History of Hot Air, and even published a book in 2014, to which some of us maybe should have paid more attention, called Pandemics, What Everyone Needs to Know. So today we're very honored to receive Dr. Doherty, who will be speaking on living with COVID-19. As an expert in immunology and an outspoken scientist, he'll speak to us about the scientific and social aspects of this pandemic, which has affected all of us. So with that, I'll leave the floor to you.
Well, good morning. Oh, good morning for me. Good afternoon for you. It's 9 o'clock in the morning in Melbourne, Australia, on a bleak day. I would love to be in Montreal. It's such a beautiful city. And I do have an honorary degree from McGill, it happens. So it would be my, not really my alma mater. They're kind of odd occasions and you don't do any work to get them. So, but it's great to be talking to you. Though I trained initially as a vet, I've spent my whole career working on problems in infection and immunity, more than 50 years. And since the Nobel Prize, I've also become a lot involved in the public communication of science. So my role in the COVID situation has really been to try to talk to people about the infection, about what it does, and about the importance of vaccination. And we do have vaccine hesitant and vaccine refusal people in Australia, as we do everywhere, I think. So I've been trying to talk particularly to the vaccine hesitant, because I think it's very sad if people catch this infection and possibly are permanently damaged or even die as a consequence of not being vaccinated. That's led me into some very interesting discussions. I actually have 99,000 Twitter followers. Which is an extraordinary experience for a basic scientist like me to be dealing with the broader public in this way. Medical doctors, of course, are much more accustomed to doing that. I had, my publishers got me into Twitter early on in the hope it would sell more books, but I had about 16,000 Twitter followers, and then I had both Google and Twitter opened on my computer. And by mistake, I put into Twitter a question about the opening hours of the local liquor store. That rapidly increased my Twitter following enormously, and I'm now enslaved by this terrible thing. But it's a very interesting and intriguing experience. And for all young scientists, I would urge that you spend at least a little of your time trying to talk about science to the broader community, both your friends and relatives, but also if you've got great visual material from the lab, if you've got a great story, try and get it out there, because the basic understanding of science in the broader community is not at a high level. People don't get a lot of the ideas that are so basic to us of probability, relative risk, and so forth. And when it comes to something like COVID-19, that can put them at risk, because they find it difficult to think it through. And that's been quite an eye-opener for me, engaging with some people who are clearly sincere, but don't seem to be able to follow the line of logic through. So the more we can all be involved and the more we can help, the better. Now, we're concerned with COVID-19. It comes from the One Health perspective, the idea that all infections and all life is kind of shared, and we have commonality with all mammalian life, particularly, and the One Health idea is sort of encompasses that. Of course, this virus has come out of bats in the wild, and somehow or other it's jumped into us. There are various speculations. One is that it went into a second species, a mammalian species in the food market in Wuhan in China, and then that spread it on. That thinking is based particularly on what happened with the first SARS virus in 2002, 2003. The virus jumped, we think, from bats into a little animal called a civet cat. You know, Chinese eat all sorts of wildlife and they buy the animals live and kill them themselves. And then it went into humans, and then it just happened to be the Chinese New Year, and basically it went all around the place. And the only place it got out of Eastern Asia, in fact, was to Canada and into Toronto, as you're aware. So that thinking, we haven't found the intermediate animal for this SARS-CoV-2 virus, if there is one. It's possible it went straight from bats to humans, particularly people who are going into caves where bats are, but we don't know. It's not impossible to prove, it's impossible to disprove that it hadn't been isolated and it wasn't in a lab and it didn't escape, but there's no particular reason to suggest it did. But anyway, what this tells us is these viruses are out there, they're out there all the time, and they're now getting across the planet much more quickly. Before 2000, we had 2 of these coronaviruses, which SARS is, CoV-2 is, circulating in humans, 2 common cold and flu coronaviruses. They cause croup and so forth, particularly in kids. But after 2000, another 5 of these viruses have crossed over into humans and spread widely. The original SARS virus in 2002, 2003, which has died out, the 2 more common cold ones, which came across in 2004, the MERS one in the Middle East, Middle Eastern respiratory syncytial virus, a virus that kills 30% of the people it infects. The original SARS killed about 10% of the people it infects. CoV-2 kills, I think, somewhere between 0.3 and 0.8% of the people it infects, but it's much more infectious. So the original one died out, and then CoV-2 came across as well. The Middle Eastern one, bats to camels to humans, and it still grumbles away in Asia and the Middle East. Unfortunately, hasn't gone any further. So that's the situation we're facing. Why do we see so many more of these infections since 2000? I think it's very much related to the fact that China has become much more prosperous, that we're seeing much more passenger air travel out of China. And that's what's getting them around the planet. So if these situations don't change, and we still in Asia continue to have live animal and live bird markets, which we worry about, particularly with influenza, it's likely we will see more of these events. So I'm at the Doherty Institute, which is named after me. I'm the patron, which means I don't have any power, but I do what I'm told by the director. And at the moment, I describe myself as the junior member on our institute's communications team. I am talking constantly, of course, with colleagues who were working with me when I was still running a laboratory, started out as postdocs, they're now full professors, they run their own program, and I look to them for the science and for telling me what's happening, and of course to the literature. Now, we've lived with pandemics and infections and a major toll of infections forever. The Black Death of the 14th century was killing 1/3 to 1/2 of the people in cities in England. And then through the next few hundred years, it regularly killed out large numbers of people in Europe. Of course, it's a flea transmission cycle from rats to us. None of that was understood. Nobody understood infection at that stage. They talked about miasmas and fogs and infection in the air, but they didn't use that sort of term. They didn't know anything about immunity. But what the people in that 14th century were relying on is what the anti-vaccination people now call natural immunity. That is, we'll catch the infection and we'll be immune. Presumably the point of that is you catch the infection and you survive and don't die, then you won't catch it a second time. That's actually true. And you won't catch it if you're dead either, because that doesn't happen. So basically natural immunity, and people who go on about that have their head somewhere else. It was a catastrophic toll of Britain. Large numbers of people died. It really broke a lot of the power of the monastics, because many of them died. There was a labor shortage. Workers demanded better conditions, and they got it for a time. And then, of course, they had to be crushed, as that's always got to happen. You can't have the workers rising up too much. People were suspected of poisoning the wells. Witchcraft was involved. It's not that different today. If you go to any bookstore, you'll see a bigger section on astrology than you'll see on science in your local bookstore, unless, of course, it's a university bookstore, if your university still has a bookstore. So these viruses come out of nature, and since we've been following them closely, they come out of nature all the time. They cause various problems. We caused the Zika virus a while back, and they cause disease issues. Some of them we've lived with for a long time. Yellow fever virus, for instance, came across to the Americas from Africa in the slave ships, and it's been active in South America ever since. We've recently had Zika in South America, where we had this terrible problem, mosquito-borne infection, babies being born with abnormalities, caused tremendous concern. That virus had actually been grumbling around in the Pacific for years, but hadn't done anything. And then suddenly it's causing a major problem in South America, causing what we're seeing now in humans with COVID, what we call a virgin soil pandemic, where you have nobody has been infected with this thing before. Suddenly people start to get infected and it spreads. Of course, you need the mosquito in the cycle to spread Zika virus because it's an arbovirus. And with COVID, like influenza, we are the transmitters. Once the virus jumped into us, it's no longer a zoonosis. We talk about zoonoses. These are infections we get from animals. Toxoplasmosis is one of them. Leptospirosis is another, that you actually get an infection from an animal. This virus has jumped into us. It's now a human pathogen. It's going to stay with us and we spread it. So we don't call it a zoonosis anymore. It's a zoonotic origin, but it's not a zoonotic disease. Now, we also, some of us may have seen the movie Contagion. It's the best movie on infection and pandemics that's ever come out of Hollywood. Usually they're crazy, but the people who made Contagion actually listened to the people who do the science, and they made a pretty good movie, except for the fact that they kind of developed a vaccine in a day, and that week they were giving it to people. And as we all know, it takes about a year to get a vaccine out there. And we got it out there remarkably quickly with COVID. That caused a lot of distress for people. Even though it's been in hundreds of millions of people, the mRNA vaccines are still described as experimental vaccines, very dangerous. Well, compared with the danger of COVID, it's kind of a ridiculous argument. If you haven't seen Contagion, try and catch it. It's quite good. Gwyneth Paltrow, you end up seeing the top of her head taken off, but it's not really her. It's a dummy. She's still around selling natural medicines and all sorts of fake stuff. So anyway, viruses carried by fruit and insectivorous bats, there's a whole lot of them. The bat immune system works differently from ours, and they can tolerate a lot of persistent infections, it seems. There are people studying the bat immune system quite seriously. Lin-Fa Wang, for instance, who's in Singapore and various others, but a small community, as you can imagine. And though it comes in that sort of One Health spectrum of comparative medicine. We used to call it comparative medicine. Now we call it One Health. What can we learn about the bat immune system that's different from our immune system that will give insights into the way our immune system works? Difficult to fund that sort of research for most animals at the moment, but maybe for bats. But they also carry other infections as well as these coronaviruses. There's lots of these coronaviruses in them. They carry the henipaviruses. These are Nipah and Hendra virus. Hendra virus is a virus that went from fruit bats into horses, into particularly veterinarians and people who work with horses. You can imagine if a horse has got a cold and it's got an infection up its nose, and then a vet comes along and lavages out the nose to give the horse some relief, you're going to get an enormous aerosol. So several people in the veterinary world died of this thing. The Nipah virus is a virus that goes from bats into pigs, and then from pigs into us. And one way it can also go directly into us is when the bats feed on the sap that they drain out of some of the palm trees. And then we also take that sap. The bats contaminate the sap, and then we can be directly infected. This is a virus that's been active not in Australia, but in Malaya, and it's got up as far as Italy, in fact, and into India. So these things move around. But these type of viruses, once they've crossed into us, they move by plane. They don't. Then it's not the bats transferring them up into India. And then, of course, we've got the filoviruses, Marburg and Ebola virus. These are almost certainly bat-borne infections that go through rodents and chimps and people in Africa. They don't just kill people. These are horrible infections. When you talk about the AstraZeneca vaccine, the Vaxveria that you've used, I think, also in Canada, as we have in Australia, the adenovirus vectored vaccine, the only time that's been used prior to COVID was actually to make an Ebola vaccine, which actually saved a lot of people in the latest Ebola outbreaks. These outbreaks happen periodically in Africa. They're not really a threat to us because these viruses are so infectious that they kill people so quickly and so horribly that we jump on them very quickly. The problem with COVID is it's such a variable clinical presentation. In younger people, it can be completely asymptomatic and people can spread it. They can have very mild symptoms and they can spread it, and they wouldn't seek any sort of medical attention if they had that. And then in older people, progressively, people can get very sick and they can die. We also know that it's more complex than that, and it's changed. In the past, we've never seen this before, but since January 2020, when we first started to live with this thing, this virus has changed. The Delta strain that's circulating now is much more infectious. It went from what we call an R0 of about 2 to 3, which is very much like influenza, in fact, to an R0 of 5 to 6. Much more infectious. It's growing to a higher titer, and it is actually causing a bit more severe disease, though we didn't really know it, didn't really work that out for a while. But what it is doing, it's infecting a lot more people. In Australia, we kept the virus pretty much out by quarantine. We're an island. We've got one national government. We're the biggest landmass on Earth, I think, with a single national government and an island state with just one government, one federal government, and six state governments, which causes some of the chaos you're familiar with in Montreal, in Canada as well, that the federal state systems and all the rest of it. But we kept it out and we were able to control it until Delta. Now Delta is established in our two biggest states, Victoria and New South Wales, two biggest populations. And it's been going up all the time. We're not going to be able to knock it out. We're heading now for 80% of the population double vaccinated. We should achieve that by the end of October, we hope. And once we've got 80%, we're going to have to open up our society. We've been locked down, and we're locked down for too long. But you can't control it like that anymore. It's been an interesting exercise, but now we're going to have to confront the virus in its wonder. So COVID, it's a disease. COVID-19 is the disease. SARS-CoV-2 is the virus. And it's a little bit, what is a virus? It's a bit of genetic material, tiny, smaller than a dust particle. You can't see it in a microscope, light microscope. You can see it in an electron microscope. It's got genetic material, and to protect that, there's some protein around it, and a bit of lipid, and a bit of sugar. And on the surface is a molecule called the spike. And the spike is the one that the vaccines are made against. The virus itself, it infects you. There's something like 29 different proteins made by this virus when it gets into one of your cells and infects you. And you'll potentially make an immune response against all those proteins. You make antibodies against those proteins, because even though they're not on the surface of a cell or the surface of the virus gets degraded and the stuff gets out. So you'll make antibodies about, there's maybe at the most 3 proteins that could be of any importance in protection, because the antibodies that protect us can't get into cells. They can only work on things on the surface. So on the surface, you've got the spike protein, you've got an envelope protein, you may be envelope and matrix proteins, a few proteins on the surface, maybe 3. So 3 of them might have some role in protection, but the principal protection with antibody is direct against the spike. But as I'll develop later, if I don't talk too much as I go through this, we can also make what we call T cell responses against little bits from all the other proteins. So what the infection gives, in a sense, somewhat broader immunity because it primes those T cells. And I'll talk about that later and go into that in more detail. So we've covered a lot of this. Viruses are obligate intracellular parasites. They can only replicate within living cells. Bacteria are cells in their own right. Many of them can replicate outside our cells. Bacteria can often move themselves around. They have myosin and all that sort of stuff that moves things around. Viruses can't. Viruses are just like particles. They're inert. They're dependent on the atmosphere, the air, and when they get into their body, they're dependent on moving in the mucus in our nose and so forth. And basically, that's the thing. It's the virus gets in, takes over our cells. The cells become virus-producing factories. They produce more virus. Some of that virus gets sneezed and coughed out and infects other people. More of the virus goes around our body and can infect us, other cells in us. And that's what a virus infection basically is. We get damaged. The virus kills cells. It'll kill cells up in our nose. People lose their sense of smell because the virus gets into the sustentacular cells, which support the nerve cells here, which are the olfactory receptors, and transmit that sense of smell up into our brain. We worry that the virus could go up those nerve cells into the brain. We're not sure whether it does. There's some sort of hints that it does. We're not clear that it does, but it certainly worries us that it would go up the nerve cells into the brain. Rabies virus does that. It goes up the nerve fibers. And because it damages cells in the nose, we can lose our sense of smell, we sneeze and all the rest of it. Then it gets down into our lungs. When we get down into the lungs, that's when we get an pneumonia. But that's not the whole story of it, in fact, because here's the interaction between the virus and the spike and the ACE2 molecule on the surface of the cell. For a virus to get into a cell, it has to attach to something on the cell. If the cell doesn't have the attachment molecule, the receptor or whatever, the virus won't get into the cell, it won't infect. So many viruses will infect only a very limited number of cells. With HIV, the AIDS virus, it infects the cells that have got a molecule called the CCR5, as the chemokine receptor on the surface. And if you don't have that molecule, the cells don't get infected. But with the COVID virus, it binds to a molecule called ACE2, which is a very ubiquitous receptor right through the body. It's in the nose, it's in the blood vessel walls, it's in the heart, it's in all your major organs, it's in your lung, it's everywhere. So this virus can get into cells all over the place. Now, the virus spike has a region called the receptor binding domain, the RBD. You can see it in the little figure, and the RBD is the receptor binding domain for that ACE2 molecule. Once it binds to that, the virus is interiorized, nucleic acid gets out, virus starts to take over the cell machinery and starts to make new virus. It's an RNA virus. The genetic material is RNA, not DNA. So the virus itself doesn't, or the nucleus is not that involved. It's more cytoplasmic. So basically, the spike protein, the way you stop the virus getting into the cells is making antibodies against that spike protein receptor binding domain. And you can see them up there, that the antibody will block that attachment. So that's the protective mechanism, the main protective mechanism and the main mechanism used in the vaccines. So over my life, I've worked on a lot of different types of viruses, all sorts of different viruses, RNA viruses, DNA virus. I've never seen anything like this virus. We haven't encountered anything like it before. It's a new and intellectually challenging experience. And it causes a very variable pathology, and a pathology that's not really like anything we've seen before. Back at the beginning of 2020, when we first encountered this thing, we were saying COVID's not the flu. But we were thinking it's a bad flu, but it's not the flu. We were wrong. And we made some wrong assumptions as a consequence early on. One of the things that was, I've been involved in influenza research off and on for about the last 30 years. One of the things we've always said in influenza is you can't stop the flu by stopping the planes. That's been kind of a mantra. It's like a religious ritual that's repeated. And basically, it's wrong. The virus, when it started out, was as infectious as flu. It spread like flu. And we stopped it in countries like Australia and New Zealand, which are island states, by stopping the planes. We stopped the planes, we stopped the virus. And so you can stop these viruses by stopping the planes. But the Chinese weren't thinking like that back in January 2020. They didn't stop the planes. Firstly, they did stop their domestic flights out of Wuhan, where the virus was first detected, but they didn't stop their international flights. We stopped them very quickly. And we didn't get much virus from that source as a result. Donald Trump stopped them in America. By the time Donald Trump stopped them, the virus wasn't coming in from China, it was coming in from Europe. So he was too late. And of course, now with the much more infectious variants, it's very hard to stop that. But if one of the things we need to do is get a global agreement that when something like this happens in a country, we'll automatically stop the planes flying out. The airline industry may not like that, but it would save them billions of dollars because they've taken an enormous hit with COVID. If we could stop the virus at the beginning and stop it getting around the planet, we'd be doing great. Okay, virus gets into us, gets into our nose, grows up there, then gets down to our lungs. What we were seeing initially, and what was horrifying the clinicians, was what we call these ground glass opacities in the lung they see by imaging. You can see them in that white stuff. That's all white crap that's in your lung that you don't want there because it's blocking up your capacity to do what the lung does, which is what air comes in, the oxygen comes out of the air, it goes into your blood, and the carbon dioxide that we make as a consequence of using that oxygen has to get out again. So gaseous exchange at the terminal bronchi, terminal alveoli of the respiratory tract. And if you've got your lung full of this stuff, it's not going to work properly. Horrible looking lesions that they saw in imaging. Interestingly, people who recover, these things are resolving somewhat better than the medics expected, which is good, but you can be left with permanent lung damage as a consequence of this. And of course, you don't want that. But the other thing about COVID that is totally different from flu is with human influenza, we rarely find any virus in the blood. It's a respiratory infection. It's a pneumonia. The virus gets into the lung. It causes damage. The innate immune response, which produces all sorts of toxic stuff at the beginning of infection, can produce so much stuff that our lungs become very leaky. They fill up with fluid. We can drown in our own lung fluid with fluid. But this is different. We've got these types of lesions. But the other thing is this virus gets into the blood and it goes around the body in the blood. So once a virus is in the blood, it's got access to every body organ. It may be kept out of the brain by what we call the blood-brain barrier, but most organs, it can get in. And it can get into the cells lining the blood vessels, which it does. It can infect cells in the heart, it can infect cells in the kidney, and it can do all sorts of damage. So it's what we call viremia, virus circulating in the blood, what we call a systemic infection. You can be a bit confused there. A systemic infection is an infection where the virus gets around the body and the blood. A disease, a virus disease with a systemic pathogenesis is something different. Pathogenesis can be something different. Pathogenesis is the sort of understanding of pathology, the understanding of damage. You can get a systemic pathogenesis when you produce a lot of these innate immune molecules in our respiratory tract. They get into the blood and they can have effects on your brain and how you feel and make you drowsy and make you sick. And in fact, when you get a sore arm after being injected with the vaccine, there's a bit of systemic effect there. We've got cytokines and chemokines getting into the blood, making us drowsy, giving us fever. This is basically completely natural. It's nature's way of telling us to slow down, in fact, because if you've got a virus infection pumping around in your blood, then you don't want to be pumping a lot more blood than you need to. You need to stop running marathons and so forth and take it quietly. So once it's in the superhighway of the blood, it can get into any cell with ACE2 on it. That means it gets into the heart. We can get heart damage. It also means it can get into the kidneys. This is why diabetics are massively at risk because their kidneys are not working that well anyway. And many of the people who die from this infection are diabetics. And it can grow in the blood vessel walls. And the roughening of the blood vessel walls caused by the virus, together with the clotting mechanisms that work through the liver, is giving us blood clots. It's what we call a coagulopathy. So it's a pneumonia and a coagulopathy. That is, we're getting blood clots. We can get big blood clots, which will cause strokes and heart attacks. And that people can die very acutely of a heart attack, particularly young people. And we can get micro clots in that blood vessels that are where the gas exchange in the lung occurs at the terminal alveoli. And those micro clots are stopping gas exchange. That's why often the clinicians initially were really quite amazed by this because they were finding that people at home, they knew they had COVID, they've had a PCR test that's found it in their nose, but they were feeling reasonably okay. Then they would suddenly crash and they would have to be hospitalized. People were finding their blood oxygen levels were way down, but they weren't noticing. They were taking it easy, they weren't noticing. And this is due to these microclots in the terminal alveoli that are actually stopping the gas exchange. With respect to one of the very simple things they discovered reasonably early on was that you can help people a lot when they're in hospital by proning them. That is, if you turn them over on their stomach, they're using parts of the lung which have been relatively spared by the infection. And so they can actually get more oxygen in themselves without having to give them oxygen. That's one of the reasons, I think, why people who are very overweight are dying disproportionately. It's very difficult. How do you prone someone who's massively overweight? How do you prone a heavily pregnant woman? Heavily pregnant women are very much at risk of this, as are their fetuses. And so it's really important that everyone, all young women, if they're contemplating having a baby particularly, or if they are pregnant, are vaccinated as soon as possible. And there's been a lot of disinformation put out about this, which has no basis in evidence at all. And I've tried to look for it, that the vaccines cause sterility and so forth. It's just a story. There's nothing there. But a number of pregnant women have gone into pregnancy without being vaccinated as a result of believing this nonsense. It's dangerous nonsense. And so that's just the acute phase of COVID. But we also have this other problem. Firstly, if you get very sick and you have to go to hospital, you're in an intensive care unit, you have to be on a ventilator, you often come out of that with some permanent damage. You've had organ damage, being on the vent and being on your back and everything, especially if you're older, doesn't do you a lot of good. A lot of people come out of this and they're never as good again. And in fact, they may die much younger. And though those deaths probably won't be recorded as COVID deaths. So there's that issue. And I think of that as post-COVID effects. They've come out of hospital, they're in the community, they're not as good as they were before. Some of them are very, very severely compromised, in fact, but some get reasonably okay. Some have permanent sort of brain fogs and dullness and all the rest of it. It's not something you want to experience. But there's also this long COVID phenomenon, and it took us a while to recognize that. It's this chronic fatigue thing that's been around virus infections for years, and doctors have had a lot of trouble dealing with it, quite frankly, because they can't link it to a cause. Here we yet often have a cause because we have people diagnosed by PCR who may not get sick. And so we've realized that people who are initially asymptomatic or have a very mild infection can get this awful long COVID, which is very debilitating. Some people it clears up. There's a very good study from the University of New South Wales where they've looked very closely at PCR positive people, they've diagnosed people, and then they've followed their long COVID. They found up to 30% of people can have some symptoms of this for up to 3 months or more. Brain fogs, muscle pains, headaches, and this sort of thing. But then with a proportion of these people, they go on and this persists, and they just don't come out of it. And some of them can actually go on. We don't know if they're going to come out of it, quite frankly. The UK at the moment has something like 1 million people registered with long COVID. It was first actually brought to the fore by patients themselves, more than by the clinical community, who started getting together and comparing notes and all the rest of it on social media. But it's a very real problem, and it's going to cause a lot of debility. It's causing a lot of drawdown on social services in countries that have a lot of social services. Sweden is one. And there are all sorts of speculations about what's causing it. We'll take it apart, we'll dissect it, and we'll find out. And it's going to give us a sort of a way in, I think, to understanding these chronic fatigue problems that people have been complaining about for years, because we have a specific diagnosis. We don't know whether it's autoimmune. Are we somehow turning on a response that's against ourselves? Is it some sort of neurological, sympathetic, parasympathetic, dysfunctional pathway? Or is, I'm very vague about all these things. Is it an immune dysregulation? What's been found by this group at University of New South Wales is that people who do or do not develop long COVID after infection can have obvious immune dysregulation in some of their innate immune type responses. We don't know, but hopefully we'll find out. And hopefully we'll find out how to treat it, because we don't know how to treat it now. But it may be the worst, in some ways, the worst part of this problem, because it infects people of all ages. It can infect little kids too. They usually don't have it too badly, and they usually come out of it reasonably quickly. But it can infect people of all ages. It infects people, young adults. In Australia at the moment, the majority of people in hospital are under 50, 80% are unvaccinated, another 15% haven't had their second vaccine shot. And so it's pretty obvious what you need to do to stop it. We don't know. We're not clear whether vaccination can completely stop long COVID. It's hard to get this information through. It certainly does reduce the incidence of it pretty dramatically, but whether we can completely stop it, we don't know. Okay, so now I've talked generally about COVID, and I'm going to say something about the immune response. The immune response is what protects us. I mean, we replicate slowly, we're large, we're complex, we're kind of like a world of microenvironments and tissue niches and stuff, where tiny organisms can multiply and invade and make us sick. So immunus comes from the Latin term immunus, which means without tax. And it was at the time of the Roman state, some of the Roman legionnaires who came back to Rome were exempt for a while from paying tax. So immunity is there to defeat the tax of infection. And we're constantly generating immune responses against pathogens that are causing mild or less mild infections and things that want to grow in and on us. So it divides into two parts: innate immunity, which is goes right across evolution. It goes into, we have common innate immune mechanism with fruit fly, and in fact, there's been a Nobel Prize for it, studying innate immunity in the fruit fly. So that's a very general, immediate type response, which can pump out a lot of rather toxic molecules in an effort to get rid of the infection or to control the infection. But it keeps us a little bit safe until the virus-specific adaptive immune response turns on. Now, that's young in evolutionary terms. It's about 400 million years old. We first find evidence of an adaptive immune system that's something like ours in the jawed fishes. And so it's about that thing. And of course comes across into the reptiles, into the birds, into us. It looks slightly different in the avian and reptilian species than it does in us. But basically, it's the same themes. It gets, in some ways, it's a case of evolution having diverged. We get to the same place, but we do it using somewhat different mechanism. Fascinating study, but I'll leave that one there. And all of the adaptive immune responses are mediated by the white blood cells. We have red cells, we have white cells. White cells are relatively rare. And it's all the adaptive immune response, the specific response, the one we generate to get immune memory from these white blood cells and their secreted products. The lymphocyte is the key cell. That wasn't even discovered until the 1960s. The last of the white blood cells to be understood, in fact. And then we have the B cells, the antibody-forming cells, the plasma cell, which is the end cell of that lineage, which makes all the antibody, sits in our bone marrow, pumps out antibody, was discovered a bit earlier. And then we discovered the T cells, the cells that interact with cell surface, not with the virus, but with changes to the cell surface that are induced by the virus. And we've known about immune memory and protection forever. We've known about it since Thucydides and the Peloponnesian Wars. They recognized at that time, they had the plague of Athens, probably typhus. They recognized that those who'd had the plague and recovered could nurse the sick, and they didn't get the disease a second time, and that it was specific. They were only protected against that. That's the basis of immunity and immune memory. And memory works by the persistence of memory B cells and T cells, which have been stimulated with the antigen, the pathogen, and are now there in larger numbers than they were in the naive state before we ever met it. And when you're boosted or when you're infected, you further expand those numbers up again. Everything in immunity and in diseases is really about numbers and where things are and what's happening in that sense. All immunity works, adaptive immunity works by protein-protein interaction. The key molecule is the antibody molecule. This is a cartoon of an IgG molecule. It's the one that's most commonly found in the blood. And antibodies tend to bind to tertiary structures on proteins. What I mean is the protein is folded. You have a sequence, a gene sequence and a protein sequence, but then it folds to make the protein. And the antibodies tend to react with those conformational determinants, we call them, the tertiary structures. Knowing how those antibodies and molecules interact, and doing that analysis by what we call structural biology, we used to call crystallography, has allowed us to design some drugs, in fact. And the initial antiviral Tamiflu, Relenza, which became Tamiflu, was designed in that way. Now, here's antibody levels in the blood. If you looked at an uninfected person, you would find nothing. And the top panel at the top left is declining antibody levels after vaccination in Israel. And you can see that the antibody levels are dropping after about, I think it's about 6 months. So the Israelis are actually now giving, you get 1 or 2 shots. They're now giving a 3rd shot, a booster shot, to push those antibodies back up again. And the Americans are starting to do the same. The bottom one shows the antibody levels after infection. So if you have the infection, you survive it okay, most people will have good antibody levels. Though we know that infected people can be reinfected, and we know that vaccinated people can get a breakthrough infection, particularly with the Delta virus. If we got a breakthrough infection in someone here with falling antibody levels, they got quite a bit of virus replicating up in the nose, then we will get a very good boost of the immune response. The bottom one is actually someone who's modeled that, Miles Davenport, a modeler, who really pretty much predicted what happened here through his modeling. Modeling can be very useful. It can give us great insights in science. And though it's basically a thought experiment, but here's the real experiment in people. So what we have in an immune response is many different antibody-producing plasma cells, the ones sitting in our bone marrow. They're all descended from B cells with a single specificity, which has and the B cell in question has a little molecule on its surface, which is much like the antibody molecule. And when the virus comes in, they get multiplied up and we get our immune response. But they're polyclonal responses. So there are lots of different clones, all reacting with that SARS-CoV-2. We've got a number of different antibodies, but they have that capacity to bind to that. And the terminally differentiated plasma cells are producing those antibodies in our bone marrow. Some years back, we also learned how to make monoclonal antibodies. What we learned to do was take one of those B cells that's going to produce a single type of antibody, not a polyclonal, but a single antibody, one cell, one antibody. That's the basic dogma of immunology. We learned how to make those, and we learned how to grow them in culture. So we can grow up buckets of a single antibody. And these monoclonal antibodies actually revolutionized a lot of the science I'm involved in because they gave us great reagents. But the other thing they're great for, they told us that in virus infections, we have some very, very rare antibodies, which will actually recognize a whole range of related viruses, like all the influenza A viruses that we have to keep making different vaccines against as they change. So those clones are there. We can make the monoclonal antibodies. We've never had to been able to make a vaccine that makes the body make a lot of those antibodies. But we do have the monoclonals, and they can be used as therapeutics. And here's one, for instance, that's been made against the original CoV-1 virus, the SARS virus, that cross-reacts with CoV-2. It blocks the antigen binding, the receptor binding, by blocking there. And basically, it can work against any of the coronaviruses. And it's being used with CoV-2. This is one that's been bought in Australia. There are other monoclonal antibodies that are very specific for the particular virus. And they're the ones that were used to protect Donald Trump when he got infected. He became infected, would have become quite ill. They gave him 8 grams of monoclonal antibody cocktail, which pulled him out of the infection very quickly. And in fact, in Florida, where you have a lot of unvaccinated people, something like 40,000 people who are unvaccinated, got infected, have been treated with these monoclonal antibodies. So the death rates would have been much higher without them. They're very expensive. A couple of shots of vaccine maybe cost you $50, $100. I'm not sure what it is in the US, but the monoclonal antibodies, by the time they get them into you and you factor in the medical costs, probably costing about $4,000 or $5,000. So it's a pretty, and you've got to catch the infection first. Now, antibodies work well against systemic infections. They're made in the bone marrow. When we're given the vaccine, it goes into our arm, goes in the lymph down to the regional lymph nodes in our armpit, these glands in our armpit. The vaccine really doesn't go any further than that in any quantity at all. Maybe a bit leaks through, I'm not sure, but I don't think so. So it's caught up in these lymph nodes, which are in the lymphatic system, our other body circulation system, the drainage system from tissues. They eventually take the lymph back into the blood, but they're caught up in these lymph nodes, which are like filter stations along the way in that drainage system. That's where the immune response happens. Cells multiply, they proliferate, they make an immune response, T cells, B cells, all this sort of stuff. And you get lots of antibody in the blood being pumped out by these plasma cells. You can get lots of antibody in the blood, but what's difficult is to keep much antibody up here with a vaccine that's given in the arm. You may get a bit more if you gave that vaccine up in the nose, and there are some vaccines, experimental vaccines and live influenza vaccines that have been given in that way. But on the main, you don't have a lot of antibody in the nose. So there's the problem. You can stop a systemic infection well with antibodies. Infections like polio, measles, and so forth, which depend on the virus being distributed by the blood. But it's hard to stop an infection up here because the antibodies themselves, which block the infection, are inert particles too. So you've got one inert particle, the virus, another inert particle, the vaccine, and they're trying to find each other, or the antibody's trying to find it if it had a brain, which it doesn't, obviously. But if the antibody finds the virus before the virus finds a cell it can infect, then you can stop the infection. But that's a very, very difficult challenge. And I doubt with any respiratory virus infection, we ever get sterilizing immunity in the nose. And of course, where we're watching COVID so closely, we're seeing these breakthrough infections in the nose. So people are getting sick. They're getting sick. Vaccinated people, sometimes developing quite unpleasant symptoms, but generally, they're not having to go to hospital. They're not going to die. And the infection is somewhat limited. So you can get infected up in the nose still, but I think when that virus gets into the blood, the antibodies bind to it, they stop it, and they stop the dissemination phase. And that's clearly what's happening. Now, all immunity depends on finding those rare cells with the right specificity and then clonally expanding them. That is, they divide every 6 hours. And if you do the math, if you've got cells dividing every 6 hours, you can get to very large numbers of cells in 6 or 7 days. After 6, 8 days, 10 days, and I'm telling you this story, it's really based on studies in mice with viruses like influenza. We haven't had time to do those sorts of studies with COVID. So part of the story of how these things work is based on what we understand from other infections. We've studied very systemically for years. Here's a lymph node, that purpley thing. That's where this all goes on. That's why the lymph nodes, you can get some soreness there. The lymph nodes are swelling up. They're pulling all these cells in. Cells are multiplying. There are pain receptors in the surface of the capsule of the lymph node. You can feel distension and all the rest of it. They multiply up. We get effector cells, and we get cells, memory cells, which will stick around for ages. So before the infection, we had maybe 1, and after the infection, we maybe have 1,000 cells with the same specificity that can be recalled to give memory. The plasma cells in the bone marrow are renewed from time to time from these memory B cells, and we can stimulate them back into action. Here's actually a slide of that for COVID, done by Ali Alabedi's lab in Washington University of St. Louis. And basically the red, these people have just been vaccinated, and you can see the antibody forming plasmablasts, the precursors of the ones that go into the blood in the blood of the vaccinated person. This is a situation, the black is the situation where they've challenged someone who was infected before and has recovered with the vaccine, and they brought them back up again there. And these are antibodies in the blood. So both infection and immunization give you loads of antibodies. But the cells I'm interested in are the killer T cells. This is a virus-infected cell. This cell is what we call a killer T cell, and it's going to kill that cell, and you can see it's killed it. It's broken down its membrane. The pink dye in the fluid that it's in has come in, and you can see that cell is really dead. So you kill that cell, it's reproducing virus, then it's also stopping the infection. So this is how you terminate an infection, with these killer T cells. Now the T cells, whereas the antibodies recognize those conformational three-dimensional structures, the T cells have a receptor that recognizes a tiny bit of virus protein, a peptide, bound in the tip of our transplant molecules. Now, I can't go into the detail on that if you're not familiar with it. Just assume that what happens with a T cell response is the immune T cell, which is programmed to actually kill cells, gets focused on the surface of the cell it needs to see by the fact that a self protein, one of our proteins, has been modified by having a bit of virus bound into it. That's all you need to think about it. And those killer C cells, when they focus on those cells, bump those cells off, and they begin the process of terminating the infection. And that's what it's about. Here's basically a structural biology picture of one of those peptides bound into the tip of one of these things. Okay, so immune response, here's basically what you see in an immune response. You see this clonal expansion of cells to the left, then they go down into memory. A lot of them die off, but there are more of them sticking around. Then when you do a secondary booster response, the numbers come way up again. Here's what's happening in the lung of an influenza virus and infected mouse. If you look at the top right-hand panels, that's the immune T cells coming into the lung to clear out the infection. They're starting to be there about day 7. They will clear that virus out between day 7 and 10. And what you see below is a secondary response when you re-challenge. They come up, they're not there immediately, but they come up sooner than they do in the primary infection, and there are more of them. And that's part of the kind of booster effect. Here you can see again, you've got the infection as sort of gray with the virus levels going up and then being knocked right down when these immune T cells come in and knock off the virus infected cells. In a secondary response, you will still get some infection in the nose, but it will be knocked down a lot faster. And that's what we're probably seeing in vaccinated people who are getting infected in the nose. These immune T cells can be protective. There's been old evidence from influenza for years that if you've got primed up T cells, you won't get as sick and you won't die. It won't stop you getting infected, but you can still clear out the virus quicker. And this is with a very severe influenza infection in China a few years back. And we did the studies in Australia, the cellular immune studies in a collaboration with the Chinese. We've got these same immune T cells operating in COVID. They're working quite well, and they're doing the job, I think, that we see. And normally when people get COVID and they get better, their virus-infected cells have been eliminated in the same way. They have all the molecules that we'd expect in these immune T cells, these killer molecules. They have names like granzymes and so forth, and perforin. And they're doing the thing. They're activated, they're killing, they're clearing out the infection. Just a couple of things. I've talked about immunity. The only thing I haven't mentioned is new drugs. There are new drugs coming through. We need drugs as well. We would love to have a pill that was cheap that you could give to anyone as soon as they test PCR positive, because the drug would likely eliminate the virus then. There's a new drug coming through from Pfizer, we believe. It's in trials at the moment. And Merck also has a drug called molnupiravir, which is a nucleoside analog. The great thing about drugs is they work across all the viruses in the class. So one of the things that could protect us against a new COVID type pandemic called by coronavirus is having loads of drugs that works across all the coronaviruses, and having drugs that we could say stockpile and then send immediately to a place where this virus infection is starting off and get rid of it quickly. But we need antiviral drugs as well as vaccines, because a vaccine still takes 6 to 12 months to develop, even with all the experience we've had with COVID. You have to do all that safety testing and all the rest of it. Okay, so it's been a bit of a gallop through this. I hope I haven't lost you too much and you've got something through it. As I say, I'm doing public communication, and I published a book called An Insider's Plague Year. I've been writing articles for our website called Setting It Straight. They're now up to number 76, and you can access them on the Doherty Institute website. Go to Setting It Straight, and they're kind of lay explainers of immunity and of the disease and of what's happening out there that I've written as we've been through this. You know, if you read them, you'd see I've been learning as we go along, and I've been changing my story a bit as we've learned more about the infection. It's complicated, it's new, it's a virus I think is a horrible virus. You don't want to get infected with it. And if you haven't been vaccinated, and I'm sure all of you are smart enough to be vaccinated, get vaccinated as soon as you can. So thank you. I'll leave it there.
Thank you so much, Dr. Doherty. We really appreciated the wonderful talk that you just gave, and also the really positive message about scientists working with the public in order to make sure that the public better understands science. I think that's something that all of us could take as scientists. And I think it's a really, really important message. So thank you so much. So for those of you that would like to ask questions, there are two things you can do. You can either raise your hand. There's a little button that says raise hand, and I will see you and allow you to speak. Or you can type your message into the Q&A section. Don't type it into the chat if you can avoid it, because I might lose it. So we're going to have about 15 to 20 minutes of questions, if that's all right. And try to keep your questions as brief as possible and only ask one question, if that's all right. So I noticed Andre has had the hand up for a really long time. So if you'd like to ask your question, I'm going to allow you to talk now. You can unmute your microphone if you like.
I'm sorry, I didn't hear what you just said.
You can ask your question. You've had your hand up for quite a while.
Oh, I just accidentally pushed it. I'm sorry.
Oh, I'm so sorry. Right.
It's okay. Well, might as well say it out loud. Absolutely wonderful presentation, and thank you.
Oh, well, thank you. I hope it wasn't too quick. I talked too quickly.
Marvelous. So sorry about that, guys. I'm sorry about that, Andre. So we have a question. Two questions from Annie Lebrec. What do you think we will do better in the future against the next pandemic virus? And what do you think will be the cause of the pandemic in the next year? I.e., where do you believe we're going to be in 2022?
Yes, this is the first really new, I mean, we've dealt with one new pandemic in modern times. There's the 1918 influenza pandemic, but that's a totally different era. But the HIV/AIDS pandemic that started in 1981 is still going. But of course, as a respiratory infection, I mean, you can make certain decisions that largely protect you from getting HIV infection, but you can't stop breathing. So respiratory infections are everywhere, and we're all at risk. So I hope we've learned an enormous amount from this. What we've seen across the planet is different countries doing different social experiments, locking down or not locking down, getting vaccine out there quickly, managing their population in various ways, providing financial support for people is tremendously important, because a lot of the people who've been killed and infected with this are people at the bottom end of the financial and social spectrum who just have to go out to work each day and to put themselves at risk, no matter what their situation, because they can't do otherwise. So I hope we've learned a lot from it. I would like to think at the end of this, we would go through, particularly as academics, and we would analyze from every aspect what happened here, using the different countries as different experiments, the different regions of the United States, for instance. The different states have done different things. Use them as the experiment. Look at it from the sociology aspect, the economic aspect, and of course the infectious disease aspect, and try to work out if there are ways we can do things better. As we know, our political systems are enormously cumbersome. It's hard to change them and to get them to operate on the basis of evidence rather than on the basis of prejudice. But we can do it if you've got good leadership. And I would like to think we might change some of the ways we do things. One of the real problems in our world is the enormously inequitable distribution of wealth, which is getting worse all the time. That's one of the things we absolutely need to change. On the purely scientific medical research front, I think it's enormously important that we develop these drugs that can be used and will work right across a particular class of viruses, because we can't really tell what's coming out next. And we're going to have more of these things coming at us if we keep up this culture of massive international air travel, particularly out of regions that have live animal and live bird markets. So we don't know what's going to come at us next. If we had drugs against all the likely candidate families of viruses, coronaviruses, henipaviruses, filovirus, maybe the noroviruses that cause cruise ship diarrhea, if they ever change, they're very infectious and they could be quite dangerous. So we could make those drugs made, we could have them tested And we could test, for instance, a drug against the coronaviruses, you could actually test by human challenge studies. You could use one of the common cold viruses and actually challenge people with that virus under controlled conditions. And once you'd established the drug itself was not toxic, which you do by toxicology studies at the beginning of any trial, and then by phase 1 human trials, where you just give the drug to see if it causes a problem. Once we had those things made and validated for, say, a human respiratory coronaviruses, we could simply switch them into treating something that came along, which is in that coronavirus class. You know, our science is incredible. With the 1918 influenza pandemic, we didn't isolate our first human influenza virus until 1933, I think it was. Okay, that's the state of the science. We made a vaccine at the time of the 1918 influenza pandemic, but it was against the bacterial secondary infection. It maybe did a bit, but it didn't stop the infection. Now, with CoV-2, we had the virus was first really reported in December of 2019. By January 15, the sequence of the virus was published with a bit of pressure from outside China. The sequence was published. And immediately, the people who made the mRNA vaccines, the adeno-vectored vaccines, were starting to design their vaccines. All they needed was the sequence of the virus. That's all. We designed a PCR test in Australia. Virus sequence published January 15th, had a PCR test immediately. First case, 25th of January, 24th January, diagnosed it immediately, isolated the virus. We were actually the first to distribute the virus worldwide to labs that wanted it. It's important to actually have the infectious virus to do virus neutralization tests, animal challenge studies, and all the rest of it. So the first thing we need to do for the future, apart from really looking at for the lessons that it's taught us, is get these drugs developed. There's a big NIH program for this. Tony Fauci has put that in place. I mean, he's a smart guy and he knows what needs to be done. Vaccines will always take some time. So it will always be a lag phase. So having drug there would be good. Would be good if we could make more of these cross-reactive monoclonals too. And I think have those stored. Though they're very expensive when you compare them with a small molecule drug. Vaccines are always a solution to infection. You know, people go on about ivermectin and hydrochloroquine. If ivermectin is a wonderful drug for treating river blindness, heartworm in dogs, strongyloides in horses, I think. It's scabies in humans. It's a great drug. But if we can make a vaccine against any of those, which we can't, it's too difficult. But if we can make a vaccine, we wouldn't use a drug, we'd use a vaccine.
Thank you, Dr. Doherty. We're getting quite a lot of questions about on the how children become infected and if they're less infectious than adults, and if the vaccine is efficient in children. So I was just wondering if you could comment on, because there's been quite a bit in the news going around about, so if you could comment.
There's a lot of debate here, and not all the doctors I think are all the responsible doctors. I mean, there's some truly raving nutters out there with medical degrees, but they're not too many, but they're very prominent on the anti-vaccination websites. But basically, the adult doctors are all pretty much agreed to get vaccinated. But with children, it's more difficult because early on, in influenza, children get infected with influenza and they die from it. The most vulnerable people with influenza are very young children who've never seen any other influenza virus because they don't have any of that cross-reactive T-cell immunity from other influenza infections, and the elderly as they get older. In 1918, though, we also saw a phenomenon we saw in 2009 with that rather mild swine flu pandemic. We saw a number of very fit young adults in intensive care and even on ECMO machines with the machine breathing for them. Very fit young adults who had these cytokine storm type problems, I think. Some died and some heavily pregnant women died. And so fit young adults are at risk, and the same is true of COVID. COVID has now moved down in the age. Maybe it's as older people have been vaccinated more. Most of the people in hospital in Melbourne now with COVID are under 50. And we've got 20-year-olds in ICU, we've got 20-year-olds on vents, we've got 20-year-olds in 30-year-olds as well. So it's moved down, partly because that top population is vaccinated. You can see it also in the data out of Israel, that it's moved down in age group. You have to look at the Israeli data in an age dissected way, otherwise it doesn't make any sense to you. Now with the kids, okay, so the kids, we're now vaccinating down to age 12 in Australia, and the Americans are vaccinating down to age 12. Adolescent boys are generally at higher risk in normal life of pericarditis, myocarditis. I think it's certainly 12 to 16, or maybe 5 to 16 even. But adolescent boys are more at risk of pericarditis, myocarditis, caused by different infections. And there's a fairly high incidence in the community. But it looks as though the mRNA vaccines are pushing that up a bit. I can't find evidence that any of these kids have actually died from it, but they've often had to go into hospital, and they've often been in 2 or 3 days and they're out again. So it's dealt with fairly quickly. But you do worry about myocarditis causing some scarring. The reliable figures, I think, at highest instance, it's about 70 per million. So there's an argument there about whether we should be vaccinating these guys. With COVID, unlike flu, the very young kids initially were not getting serious symptoms. In the main, there were a few who got a horrible inflammatory disease we call Kawasaki disease in the past, but a few of them got that, but very rare. And so mostly they were pretty okay. And when the virus came into the household, often the kids either didn't get infected or they didn't show any symptoms. So we thought, and they weren't bringing the virus into the household. Now that's changed. With the Delta virus, once the virus gets into the household, if you test the family by PCR, you'll find everyone gets infected, including the little kids. They'll all get infected. And we also know from numerous instances now that the kids can bring the virus into the family. So, but you wouldn't vaccinate the small children just to protect adults. So the question is the possible side effects of the vaccine problematic enough so that you rather have them have COVID? Well, they can get infected and die of COVID. It's a much lower instance. And we had basically in the US, we had the heads of all the pediatric hospitals saying, we've got a real problem. Our ICUs are full up. They had 2 problems simultaneously, actually. They had COVID and they had respiratory syncytial virus, which is a problem of babies. So these 2 viruses, but that can happen anytime with kids. There's a lot of respiratory problems caused by different viruses, enteroviruses, paraflus, respiratory syncytial. So the kids are at risk. So personally, if the trials all go through fine, they're currently in progress, if they look fine, if they look safe, if it looks protective, I'd personally be in favor of vaccinating kids, but it's going to be up to regulatory authorities to approve, pediatricians to endorse it, and parents to accept it.
Thank you. We just, Levon, you've had your hand up for quite a while, so I'm gonna allow you to talk if that's okay.
Can I say something?
Oh, yes. Sorry.
Yes, Peter, thank you very much. It was a wonderful lecture, and on behalf of the student, thank you. I have a question. In the long COVID, the patients are mentioning that they have a lot of chronic fatigue.
Yes.
I wonder if the chronic fatigue syndrome or polymyalgia has something to do also with some virus infection.
Yes, Michael, we've long thought that viruses are triggering chronic fatigue and polymyalgia and all this stuff, but we've never really been able to link it back to the virus. And we thought, for instance, Epstein-Barr virus is a very ubiquitous infection, could be one of the causes. But it's been extremely frustrating. And it's been frustrating for the patients, firstly, to convince the doctor that it's not psychosomatic. And secondly, for the doctor to realize that this person is really suffering, but they don't know what to do for them. And it's been a very problematic situation. But now with COVID, because we've done so many PCRs, as we've done contact tracing and so forth of people, we've got all these cases where we've had an asymptomatic infection or very mild infection, which we would never have detected, but we know it's been caused by SARS-CoV-2. And then we have this subsequent problem developing in a month or 2, which is clearly causally related back to it because of the incidence as much as anything else. And we don't understand the mechanism, but this is going to provide a great opportunity to open that door to analyze these chronic fatigues. We know in some people there's evidence of cortical shrinkage in the brain. We think there's all sorts of possible disrupted neurohormonal or neurophysiological pathways. We don't know. And of course, it goes across the whole expertise of medicine. The neurologists, the infectious disease people, the immunologists. The immunologists particularly ask, well, is the virus persisting in some way? Is that driving it? Or is just some of the virus, the viral protein, is that persisting in some way in some site? And that we're looking at the immune dysregulation. But a lot of other clinical, we need a lot of other clinical insight from endocrinologists, neurologists, all sorts of people, as we try to pull this apart and hopefully find what are the underlying issues and hopefully develop useful treatments. So hopefully we can open out that whole syndrome. The other issue is, of course, is there autoimmunity? And the other issue, of course, with all these virus infections, we've long suspected that many autoimmune diseases are triggered by an infection years previously, but you never can get back to it because you don't know what that would have been. With COVID, we'll have all that record of these people who were infected. And maybe if they later develop a higher incidence or an unusual autoimmune disease, we can tie it back.
Thank you.
Thank you, Dr. Doherty. So Levon, if you'd like to ask your question now.
Thank you. A very interesting talk. I'm Levon Abraham. I'm from University of Montreal, virologist. I have two short questions. First, do you think that this asymptomatic development of a disease in or just light symptoms in kids is due to the protection by maybe our respiratory disease? And second question is, do you think this SARS-CoV-2 and COVID-19 will become eventually a common cold disease for our population? Thank you.
Yeah, thanks. Good question, Levon. I mean, you're suggesting that the fact kids are having these other respiratory things, as with anyone who's got a small child in a childcare facility knows that they're a constant source of infection, and that infections we had as children don't protect us from being reinfected by our own kids, and they certainly don't protect Granny in the nursing home who would have had the infection as a child, then the infection from us when we were growing up. So yeah, but could they be sort of stimulating some sort of non-specific innate immunity and giving some protection? Possible. We do think it's something to do with interferon pathways. That's part of the thing. But the thing about this virus is it's behaved like no other virus we've seen before, and that is that it's become more infectious. And I think that's just that it's been in a virgin soil situation. The Delta virus is first detected in India in December. They started giving some experimental vaccines in November, but it's pretty unlikely, I think, that the virus has been selected to jump the immune response. I think that Delta is just a virus that grows faster and grows to higher titer, and therefore it's taken over because it infects more. And it's infecting more children, many more children. So even though the disease is maybe a bit more severe in kids, we're actually seeing more kids infected because, as with adults, it's more infectious, so more are getting clinical symptoms. So it's the situation in kids and what's happening there is still not clear, but there's something about that pediatric immune system, I think, that's handling it better.
Thank you, Dr. Doherty. So now there is a question which we feel is quite important. It's how can I, as a citizen scientist, speak to others about the importance of vaccination against COVID-19, and what message will get through?
I wish I could answer that. I've been trying very hard. I think people get fixated. Well, there's several things they get fixated on. One is this thing, natural immunity is best. You know, there's all this stuff out there that says natural is best. And you know, basically when you're eating food, I mean, you know, it's great to eat good food that's not grown with lots of chemicals, because I wonder about all these chemicals we use in the environment actually and what they do to us. But basically when it gets into our stomach, it's just chopped up into bits and it goes across into our blood. I mean, food's food. So natural immunity, that's the buzzword for the anti-vaxxers actually. Get the infection. And, you know, it doesn't make any sense to me that if you're trying to stop yourself getting an infection, you first get the infection to stop yourself getting the infection if you survive. That argument doesn't make much sense. I think people get off onto these things for all sorts of reasons. They get convinced they've got special knowledge, or they listen to some person they regard as influential, or they believe they're being brave and unconventional. And all I can try and do is try to get them to think it through, and to think through the fact that if nothing is without risk, that the risk of actually dying in Australia from the AstraZeneca vaccine, which causes this horrible blood clotting problem, which is unique to that vaccine, is about 1 in 500,000. And with a few more being actually probably damaged in the long term by getting this clotting problem, but surviving. So 1 in 500,000 dead. Your chance of dying in a motor-related accident, a car-related accident in a year in Australia is 1 in 25,000. So these people who won't take the AstraZeneca vaccine are not avoiding getting in cars or crossing streets or going on the pavement for obvious reasons. We have to do these things. But it's very hard if you can try to get people to think in terms of relative risk, but it seems very hard to get people to do that. And and there's also a myth out there that this virus, either it doesn't exist and it's a plot. I mean, this is a vast plot between all the medical profession, the governments, the drug manufacturers and Bill Gates, who started the plot. I mean, Bill Gates wants to put 5G chips into all of us and all this sort of nonsense. The craziness out there is almost impossible to contemplate. We've had demonstrations here, and we had some feedback that some of the demonstrators who are anti-vaccination were thinking the police had put the vaccine in pepper spray. That's an interesting approach. So try to talk to them about the disease versus the virus.Ry to tell them what the disease is like and why it's a bad disease, and why, if they're worried about getting a clot from vaccine, they're 800 times more likely to get serious clots from the infection. I mean, it's getting the people to make that step. Now, we're trained to think like that. We're trained to think in terms of probability. We're trained to think rationally through a problem, dissect the problem and take it apart. Scientists like me that are highly successful, what we do is we always, when we get new data that's uncomfortable, we always try to take it back to first principles, break it down, build it up again. Something is very difficult to do in politics, but you can do in science, in the lab. But people don't do that. Yet if you can get them to break it down to the basic principle, and start not from the vaccine, but start from the virus. They don't understand infection. They don't get it. And I don't know why. And I don't know, maybe in the next life, I'd come back as a psychiatrist rather than a dung beetle, but who knows?
Thank you very much for that very insightful answer. And thank you to Rosa Caparucci for asking that. We have one final question, and then I'm going to give over to Sophie to close the show. And this final question is from Mariam. So I'm going to allow you to talk now, if that's okay, Mariam.
Hello. Hi.
Right.
Hi. So thank you for your interesting presentation. My question is about long COVID. Actually, we're working on long COVID. My question is that we know that the vaccination can prevent severe acute COVID-19, but is there any data about vaccination preventing long COVID as well?
Yeah, Mariam, I've been looking for that too. I'm hoping if you were working on it. It's kind of, there's real complexities with long COVID. Firstly, there's case definition. What are we describing as long COVID? Secondly, a lot of the information out there is self-reporting. Now that's fine, but you really want series that are being studied by professionals who are really looking at the disease mechanisms and bleeding people and all the rest of it. So there's one study in University of New South Wales that's really good that's doing that. And I'm sure there are others across the planet. So how well does vaccine prevent long COVID? I'm getting all sorts of sort of answers back, which none of them have got really good solid basis in data. And it goes from saying, well, I'm running a long COVID clinic and none of the people have been vaccinated, to saying, well, it reduces the risk of it 50%, but nothing about severity, duration. I mean, this information will come through. Have you actually got a clinical trial going on where you're looking at people with long COVID? And have you picked up on any who've been vaccinated and been infected?
Yes, actually, we're doing that.
What do you think is the incidence in the vaccinated people?
Well, so far, our patients mostly had the long COVID for more than 3 to 4 months.
Okay.
So most of them were not vaccinated when they got the disease. That's why it just came to my mind that it could prevent long COVID. So far, we didn't have any patient who was vaccinated and then got the long COVID.
Yes, I see that. That's great to hear, because that's what I want to hear and what we all want to hear. But it's until guys like you who are writing up sequential studies start to publish stuff, we won't really know what's happening here, I think. There's possibly an unexpected paper out there that's due to come out, but I haven't seen. Have you seen anything published that's serious on vaccination preventing it?
No, not yet.
No, we're all looking for it, I think. But part of the problem is case definition. And what he's saying, when it prevents against long COVID, what are people saying? It prevents against long COVID in those who are symptomatic, or it presents against long COVID in those who have breakthrough infection, because many people who get a breakthrough infection or vaccinated, I expect, won't show any symptoms at all. And you know, early on last year, we were picking up reinfection in people who've had the infection because they were required to have a test as they went through an airport or something. But so I suspect that most of the people who've been vaccinated aren't at much risk of it. But I wait to see really good data, and I'd love to see great data on it.
Thank you so much. And I'm really sorry, everyone, but I'm going to have to close the show. So I'm going to pass on to Sophie, but your questions were all brilliant. And thank you so much, Dr. Doherty, for taking the time to answer so many and so well. Thank you very much.
Thanks.
Yeah, thank you. So again, thank you, Dr. Doherty, for this enlightening talk with really clear and informative science on such an important subject. I also wanted to thank the Academic Affairs and the Communications Department of the RCM for all their great work in the organization of this seminar. And to thank Dr. Chrétien and the Marie-Louise Chrétien Funds for allowing us to bring such great science and such prestigious scientists to the RCM. Unfortunately, not in person, but soon maybe. And finally, I wanted to thank all the people in the audience for attending this talk. And if you're ever interested in attending more seminars, you can look up our brand new website, which has all the relevant information. So rcm.qc.ca, and follow our Twitter. As Dr. Doherty said, we're pretty active on Twitter because it's important. So at IRCM _MTL. And with that, thank you and good evening or good morning.
Okay. Well, thank you. It's been a pleasure to talk to everyone and to interact with you, and have a good evening in Montreal.